Hello. This is a video tutorial or a video overview of the culturing exercise that we did for the course weekend. If that's okay, it's going to be in English, simply because it's a bit easier for me to sort of communicate exactly what to do and how to take care of safely all the materials that you're working with. So I thank you for your patience. Yes, so the culturing exercise, vi odla mikroberna på och runt oss. So, if you have not already, you should have downloaded this course exercise from the Canvas website in the module for Digital Kursträff, Hemmalab, instruktioner. And if you have received your kit, which you should have, you will have this brochure in it. So you should have read over this already, as well as the instructions here. And you should therefore have prepared a few materials ahead of time before starting this exercise. So if you have not done that, after we go through the materials you should have, stop the exercise and go get them. And then things will go a lot smoother for the actual exercise. To start off with, just leave the plates closed until you're ready to use them. And don't touch the media with your fingers or hands or anything like that. We want to avoid contamination. And we just want to avoid any exposure to the chemicals that are in a couple of the media items. I'm going to point those out. So after working with the inoculating materials like the blue plate spreaders and the wooden pins, put them directly into a waste bag and don't touch any of the media that might be on the pins, just to make sure that you keep that separate and thrown away. After the plates have been inoculated, you're going to seal them with this stuff that I've included called parafilm. I'll show you that. And then after you've performed the exercise, all materials, just put them in brännbart, just to dispose of them properly. So that will be the first day, and then you'll incubate the plates and the agar tubes overnight. And then the next day, you just leave everything closed, and you can make your observations through the plates. You can just leave them all closed, and I'll show you what I mean in a minute, but don't open anything, just leave them closed. You can perform the observations that I've asked for in the exercise. And then when you're done with the observations, you can dispose of the plates again in brännbart and the tubes. So the first thing you'll need to make sure is you have a clean surface. And you will also need to have handsprit and some paper towels. So in addition to that, so that's to start with everything being clean. You will also need a container for waste. So I have here just a plastic bag in a beaker. So you can have any small container, but make sure you put a plastic bag in it, because that way you can remove it and throw it into brännbart. You should have already had an overview from me about what to do with all the materials after they've been used, but I will reiterate, everything goes into brännbart. So that includes all inoculating materials and the cultures after they have grown, after the second day of incubation. So now make sure that that's set. We'll have this to the side. And you will also need to have on hand, from your own supplies, a pair of scissors and a marker, and you will need some samples. So you will need a soil sample. That's the first item on the list. This is just ordinary soil that I got from actually potting soil. You will need a water sample that could have been taken from anywhere, a pond, a ditch, lake, but you'll need your water sample. You will also need, and you should have collected these in the sterile Falcon tubes. This is called a Falcon tube, by the way, 50 milliliter plastic tube with a screw cap. You should collect your samples in these items, and then you should have one empty that's left over and keep it closed because it should be sterile. All right. So you need to have those. And in addition, you should have food items for culturing. I don't have any now, but for an example, I have one food item. So this is our fermented food item. We'll just call this fermented food item. In fact, I'll even write it down. Fermented food. Ta-da! Here we go. So that's one of, I believe I suggested up to 4 or 5 different food items you can try. And that will be in the second part of the third part of the culturing exercise, rather. So make sure you have at least 4 different items. I think I suggested natural yogurt, Activia, Yogi. You can even use baker's yeast, sourdough starter. I think I even at the last minute suggested you can try kombucha. If you have not sparkling kombucha, but stuff that has been actively fermenting. So you have organisms growing in it. That could be interesting. And you should also have a pot of, I have a beaker, but you should have a batch of sterilized water. So this water should have been boiled for about 15 minutes. I believe I wrote 15 minutes down. 10 minutes is fine. And then cooled to room temperature, but remained covered so that the water is sterile and nothing is fallen in, no dust. So it should be sterile. And we'll have that to the side here for now. And that should be about it that you will need. Oh yes, and a couple more things, sorry, almost forgot. You'll need a kitchen scale. I have a bit of a fancier one here. So you'll need a kitchen scale and something to weigh the soil with, or rather extract it or scoop it up with, a teaspoon. So we'll put that over here for now. So those are the items you should have. And from your culturing kit, also make sure you have on hand a package of four what are called trypticase soy agar plates, TSA. These have a red stripe on the side, and leave everything in the bag until we're ready to work with them. Excuse me. And a bag of potato dextrose agar plates, so PDA, and they have a blue stripe on the side. And remember, as we talked about in person, this one is the one that has the antibiotic in it, the chloramphenicol. So again, don't handle the media with your bare hands. It's not dangerous, but just to be on the safe side, if anything spills out of this on the floor, whatever, wear gloves, pick it up with paper towels, throw it into the Brembart, and then just wipe it down with a lot of water, throw that into Brembart, and you'll be fine. It's not really not anything not anything toxic and harmful. It's toxic to the bacteria, but at these concentrations, really not to us. So we have these 2 plate media, but you should also have 6 of these orange colored tubes, and these are what are called agar slants. So agar is the gelatin. That's what solidifies the media. But you can see it's in a slanted form. And as we discussed, that's because you have one section. Well, I'll go into that in a minute, actually. But this also has another compound in it, the phenol red. So if you drop this on the floor and it breaks, or if the media falls out, again, pick it up with a pair of gloves. Don't handle the media with bare hands. And then throw everything into Brembart. You will not be harmed by this, but it's just to be, again, on the safe side. So those are our media components. And you should also have 2 packages of These are sterile plate spreaders. So they have a little L form and they're pre-packaged and sterile. And you should also have a tube that has sterile pin nut. And these are what we'll use to inoculate the tubes. And I'll show that in a minute as well. You should also have parafilm. So this is what we use to seal the plates with. And all it is sort of a wax film that can stretch out. I've probably demonstrated it to you, but it's a wax film that stretches out and it can be used to wrap around the plates and seal them up. And finally, you should have a bag that will have three sterile pipetters, plastic pipetters. And we will use these to transfer our samples onto the plates when we do the plate culturing. Okay, so that being said, I think we are ready to culture. So I'm just going to move these to the side. And the first exercise is to culture the soil and pond water. So what we will do first is we will sterilize the bench. So take a bit of handsprit, put it on your table, on your bench, wipe it down. Make sure it's good and sterile. And this can just go in the garbage, of course. It's just our paper towel, or you can hold on to it because you might use it later. And I'm also going to sterilize my hands. Okay. Excuse me. So now we will take the plates out of the package. They're in there fairly tight. So all you have to do is just pull on the corners a little bit and just gently push them out. There we should have our plates. Now what I want you to do though, before we work with them, is check each of them to make sure there is nothing growing on them, to make sure they're not contaminated. And we also take out our TSA plates. Okay? And we'll do that in the same manner. Just remove them from the package. Nice and easy. These have no other compounds besides nutrients in them. So these are perfectly fine to handle, but leave them closed anyway because we do not want to contaminate them. And again, double check that you have nothing growing on any of the plates. Again, if you see air bubbles, that's okay. But if you see like small little dots that are whitish or irregular in their boundaries, if they look like colonies, and you can go back into your exercise and check what a colony would look like, you're going to want to not use that plate, unfortunately. So you might have to, if they are contaminated, you might have to skip an exercise, but I don't think you'll have any, I hope. So now that we have our plates out, we can have our inoculating sticks to the side and our parafilm. And then let's bring over our first sample. Oh, and our sterile pipetters. So we'll have those. So let's bring over our first sample, and this will be the soil. So what we're going to do is we're going to make a soil dilution. Now we're going to need the sterile water for that, but we make a soil dilution simply because if we were to just put soil directly onto the plate and just sprinkle it on, there's two things that we do not know. Well, two problems with that, rather. One, there's probably a lot of bacteria in here and it would completely overwhelm the plate. So we want to actually see distinct colonies the best we can. Another more scientific reason that we want to actually weigh out a gram of soil into the fresh tube and then fill it to 50 milliliters of water is because we've then made a 1 in 50 dilution. Now, practically we can't count the colonies because it's still going to be too dense. But the thought is, the scientific reason for that if I can explain here, is that, let me get my marker. When we have a tube that we then put 1 gram of soil in, and then we fill it up to 50 milliliters, of course, that is going to be, because 1 milliliter is 1 gram, we have 1 and 50 dilution. So if we were to then take out of this, let's say 0.25 milliliters, and then we put that onto a plate, and then we were to count the colonies, and like I said, we're not going to do this because practically it doesn't really work and we don't have enough room for the material to send by the lab. But the idea is you can count the number of colonies on the plate, and then you know that that number came from 0.25 milliliters. So for every 0.25 milliliters, let's say you have 100 colonies. You can then back calculate that 0.25 milliliters of a 1 in 50 dilution. You can then calculate the number of colonies that are in 1 gram of soil. So that means that by making dilutions and making them exact dilutions, we can also quantify the number of colonies or the number of bacterial cells, potential bacterial cells. We actually call them colony forming units, CFUs. We can get an idea, if we were to compare different samples, we could get an idea how much bacterial mass or microbial mass was in a gram of soil in one soil sample versus another. So you can use these kinds of methods for quantifying bacteria. This is still done on occasion. We use molecular methods more now, but some people still will take and make dilutions in order to count the number of bacteria in soil. So let's start making our 1 in 50 dilution. So we're going to need our scale, and we're going to need our sterile tube, and you're going to need something to hold the tube on the scale. So what I recommend is that you take a small little cup. I have a beaker here, and you put the tube in the beaker and put it on your scale, and then you tare the scale. So now that you have that, you can then go in and get your 1 gram of soil. I'm gonna have to, this is a bit of a big spoon, so I'm gonna do like this. So I'll put that in carefully. So that's a 1/2 gram, a little bit more, until we get to 1 gram. That's close enough for this exercise. We're usually a bit more exact, but we can forgive ourselves this time. Okay, so we will now take and our sterile water. You can transfer now sterile water to the tube and fill it up to the 50 milliliter mark. I'm going to pour it in, but if it's a bit difficult to pour, use one of your sterile pipetters and just transfer it over. Just for the sake of doing that and showing you what I mean, to make sure it's clear, I will do that myself, because it might be difficult to pour from a pot. So take out your sterile pipetters, just don't touch the tips, just touch the ends. The bulb, this is the bulb. And then we'll just transfer over 50 milliliters. It might take a little while, but we'll do like this. So that's what I mean. If it's a little difficult to pour into the pot or pour from your any pot that you use to boil the water in, you can just transfer it like this. It only takes a little bit longer. And keep the tip out of the tube so it stays sterile, so you don't get any soil on it or any splashing on it. Almost there. And just about there. Just a bit more. Beautiful. Okay. We'll put that back on. Put this to the side for now. Just try to keep it a bit sterile. So now you have your tube with your soil and your water. Close it. It should be at 50 mils again. And we're going to shake it because we're going to try to break apart any soil particles and also see if we can knock off any bacteria from any clay particles or other soil particles that are in your soil matrix. This soil actually isn't breaking apart so well. So we're just going to have to accept that, but we'll see if we can really beat it up. It's okay if it doesn't break apart fully because you'll still get plenty of bacteria, but ideally we would have a very well homogenized soil sample. So that way we really get an even mixture of bacterial cells mixed around in the water. So do that for about, I would say, 1 to 2 minutes. And like I said, this is a bit suboptimal, but we'll accept it anyway for now. But under ideal circumstances, it should be completely homogeneous. But now that we have that, our soil sample, our soil dilution rather, can move that, the scale to the side. And now, if we still have our sterile pipetter, we can do the plating. So this is quite simple. You are first going to take a TSA plate, and that's the one with the red stripe. You can see here. And remember, this is our trypticase soy agar. This is a very general media with no antibiotics. So this is what we're going to use for the bacteria. You might get yeast and fungi growing in it, but the bacteria will grow, typically they will grow quite a bit faster. So those would be the first things you see. You might see a couple of yeast cells though, or yeast colonies rather, because they also can grow quite fast, but I doubt you'll see much fungal hyphae. So when you are ready, leave it closed, sorry about that. And you have your plate spreaders and open them up, and you open it up only part of the way. Don't open it all the way. It should be just open to there, so that way this is closed, this part of the bag, and it stays sterile. So when you're ready, you have that. Now, take your pipetter, open the plate, take your sample, get out of the way, and you will take, you can see on the pipetter that there's gradients on it, there's markings, and they are every 1/2 or 0.5 milliliters. I want you to take 0.25 milliliters. So just up to that level. Maybe you can see that there. So it's just halfway up to that very first mark that's on the pipetter. And then you're going to put the drops on the plate directly. It should be between 3 to 4 drops. 1, 2, 3, 4. And that should be plenty. Put it to the side, your pipetter, because you can reuse it because you're going to inoculate the other plate. Close. Take out the plate spreader, and only don't touch that part because it has to stay sterile, just the end. Then all you do is spread it around. You make sure that you cover the entire surface of the agar plate. And you can do this until you feel like you've gotten it nice and spread. Okay. Close the plate. And this goes directly into our waste. So that's one plate. And now we can do the other plate as well. This is the PDA with the antibiotic. So this is for the fungi. I should mention that you're probably wondering, why don't you just reuse this if you're just using the same sample? Well, the thing about media is that if you have different media for different things, you don't want to transfer any potential nutrients or compounds that might be in one media to another one, or vice versa. Sure, you might have the same inoculum, but the medias themselves have different compositions. And even after touching the media, you can still have trace amounts of different chemicals or nutrients that you don't want to transfer to another plate. So whenever you work with plates, you always work with a sterile inoculating spreader or inoculating loop. Every new plate should always be handled with a sterile instrument. Of course, it depends on the application, but in general. So we'll open up our PDA plate. We will again take another 0.25 mils. So that's halfway up to that first gradient on your pipetter. And 1, 2, 3, 4 drops. It should be about 4 drops. We can actually get rid of that in the garbage, the pipetter, because we will need another sterile one for the next exercise. A new spreader, and then we spread. Again, you don't touch the auger, you just touch the side of the plate if you need to move it around. Spread it nice and gently, nice and evenly. Take that into the garbage, close the lid, and now you have your auger plates with the soil sample. So what we should do now is seal them. So get your scissors, get your parafilm, and then your marker, because we're going to want to label it too. So for sealing plates, we use the parafilm. What you can do is you can cut a strip that is about, I would say, maybe about 1.5 centimeters. Don't use too much because you only have a limited amount. So really no wider than 1.5 centimeters. You know, that's about the front knuckle of my index finger. Pull off the backing, gently pull off the backing, so you don't rip it in half. So you should still have one continuous piece like that. You pick up the plate, put one end under your finger, like that, against the side of the plate, and then gently, gently pull until you feel it's just enough. It'll start to not pull out anymore. You'll feel it. It'll start to stop. And that's when you want to stop pulling, because otherwise you'll rip it. Gently pull, wrap it around, and keep going until you get to the very last little bit. Push it closed, and you should see that the parafilm covers the whole side with a little bit of a lip on the top and the bottom of the plate. So that is a sealed plate. We'll do that with the PDA plate. Well, that was the PDA plate. We'll do it with the TSA plate. You can throw these in the garbage as well. Wrapped, and there we have it, our soil sample. Now, you'll notice that what I did is these right now are right side up, so the plates are like that, so the lid is on top. What I like to do is flip them upside down, because you're going to incubate them upside down. But you can flip them upside down and then label them. I'm going to label this soil. And I'm going to put the date. So what's today? Today is the 25th, right? So let's see, that's 25/03/21. And because you have the colored stripe, you'll know which plate is which. And why do we incubate them upside down? Well, when you have them right side up, sure, you'll get colonies that grow. And in a lot of applications, you can grow them right side up like this. But incubating them upside down actually helps you get better formed colonies, because if I can get this, what happens is the colonies, if you have your plate growing upside down, the media is like that, then here's your lid, right? Well, that looks like it's right side up to you. No, that is upside down for you. A colony will, because gravity is pulling in that direction, right? So a colony will form like that, and you get much more distinct colonies, because if it's right side up and gravity is pulling like that, sometimes the colonies can be a bit smushed and they can be a bit flatter, and they're a bit harder to discern and distinguish between the two. So we like to incubate them upside down. So those are our two soils, or our one soil and the two different plates. So we put those aside, and we're going to do essentially the same thing with our water sample. This is easier because all we're going to do is use the same amount, 0.25 milliliters, and then inoculate each of the plates to see what grows from our water samples. So for this, you'll need another sterile pipetter. So take out another sterile pipetter. We'll take the first plate here. This is our TSA plate. So all we have to do is open it up, open the water sample, take out again 0.25, because we also kind of want to compare the number of colonies that we see between the soil and the water sample. It'd be interesting to see which one has more in it after we've made a dilution. But we take a half halfway up to that first gradient on the pipetter and drop it onto the plate. 1, 2, 3. That was 3 this time, so I think I had a little too little, 4. Put that to the side for now, our pipetter. Go back and get another sterile plate spreader and do the same as we did for the soil. Nice and even. Into the garbage, close, to the side. Now let's do the other plate. So this is the PDA. This is the potato dextrose agar. 0.25 milliliters, halfway up to that gradient on the pipetter. 1, 2, 3, about 4. That was about 4. We'll call that 4. But I'm pretty sure it was at the right mark. Put that down. Get one more plate spreader and spread that around. Nice and even. Into the garbage and close. And we'll seal that up with our parafilm. Push it down and so it sticks to itself, and then you got a perfect seal on the plate. Put that aside and do the next one. This is our trypticase soy agar with a red stripe. Oops, little rip. That's not bad. Just pull it a little bit. There we go. And we'll call this water 250321. Okay, so there's our water sample. Now there's one more thing we should do. You can put that away. Is we should do a control. And this is for the sake of the soil, but it's good to check the plates too. But what we're going to do is we're going to take our last sterile pipetter. We're going to see if anything grows in your water. So how well have you sterilized your water and kept it sterile? So we're going to do the same thing that we just did with our pond water. We'll move that to the side. And now we'll just simply make negative controls, which are our sterile water. So if we've kept everything sterile, you should not see anything growing on here. So we're going to go in and get 2.25 milliliters of water, sterile water, drop it on. Oops, that was the wrong lid. I think I probably just contaminated it. Anyway, don't do what I just did. But go back, get some new plate spreaders. So remember, open the bag, take one out. Now, spread the water around. Into the garbage. Close that. And then do the same with the other plate. Okay, so you can, this actually, because you use it with sterile water, you can save this one if you want to use it for later on with your microscopy exercise as well, because this should be clean. Even if there's a bacteria or two growing in it, it's fine for non-sterile use. So you can save that one if you want. Another plate spreader and spread it around. There. And we'll seal these plates up as well. Notice I just make a little rip in the paper backing on it. Take our second plate. Stretch it out nice there. And there you have it. So I'm going to put negative control. 250321. And why negative? Because we should not get any growth on that plate. We'll put those to the side. So that was our environmental samples, and that is part of exercise 1. So jord vattenkultur. And now we're going to have a little fun to see what is growing on you. Because of course things are growing on you. We're covered in microbes. Unfortunately, I miscounted, and there's only 2 plates left for this exercise. But that's okay, because what we'll do is take your marker and draw a line down the center of each plate. Okay? Then we have 2 halves of each plate. You're going to label one side sterile and the other side, we'll just label it skin. So we'll do that with the other plate. So all we want to do for this exercise is to test out how good your hand sanitizer is. So we're going to inoculate one side from our forearm here and see what's growing here, and we'll inoculate each of the plates. We will then take our hand sanitizer, and then we can do the other side, and we can sterilize the skin and then see what we find growing on that after it's been sterilized. So I'll walk you through it. So take out one of your plate spreaders. We'll put this to the side for now. And one plate. Keep in mind which side is the skin side after we flip it over. Take out one plate spreader. I'm going to run out of plate spreaders here. Take your arm and just rub it back and forth. Put a little pressure on there just to try to see if you can really pull up some microbes, and flip it around a bit, but really rub it around there. Okay. Skin side here. And just keep to this side of the line, the skin side of the line there. Don't go over, don't cross over. You can flip it over if you want to, but just inoculate on this side of the line. Into the garbage. You can put that other plate to the side. And we'll do the same with the other plate. So once again, and then just inoculate the plate. Okay, so now we have our skin sides inoculated. We will now sterilize our other arm and inoculate the other side of the plate. So with our hand sanitizer and a paper towel, just get these out of the way. We'll put a little bit on there and just wipe the arm down real good. Okay, put that in the garbage there. So now we have our sterilized arm. Take out a plate spreader. I'm running out of plate spreaders, unfortunately. So I'm only going to do this once on the video, but you should have one more left. I only have one left, but you should have one more of these left. So for now, take the first one, the first plate spreader, rub it on your arm, and take the first plate. Make sure you find the sterile side and inoculate that side with whatever might have survived. Okay, take that, throw it away. Now repeat for the other plate, but use a new plate spreader. I don't have one with me. Yes. Wait a minute, I do have some. Haha. So we'll take out a new plate spreader, sterile one. Rub that onto our sterilized arm. And then we inoculate the second plate on the sterile side of the plate. Yeah. Into the garbage. There we have our exercise 2 to see how well your hand sanitizer performs. And we seal these up with paraffin once more. As so, there's one. That's our TSA plate. and the second one. So our skin plate, our PDA plate with our skin samples rather. There we have it. So now that is the second exercise that we did. And you should have now, just throw that away. You should have now a set of 8 plates. So 2 controls, 2 water samples, 2 soil samples, 2 soil plates rather, and skin plates. The red again, remember that's the bacteria. So those are our trypticase soy. Put those together. Hello, where are you? There you are. There. And we're ready to incubate. So again, make sure that the plates are upside down. So we incubate them upside down. And you are to put these in a darkened cabinet, at least at room temperature. If you have a warmer place in your house, then you can use that as well, but don't go above 30 degrees. Keep it around between 25 to 28 degrees, or even like 22 degrees, that's fine too. But a little bit warmer will help you grow faster, will help the colonies grow faster. But keep them out of reach of children and pets. So away in a cabinet where no one else can get at them but you. Okay, so we'll put those aside for now. And that is the first part of the exercise. And we'll just go briefly through the second part, which is to determine whether or not you have different types of fermentation going on. So in your instructions, if we take out our instructions, skip ahead a little bit to this table that you see here. I'll just give you a quick explanation. These media are used as indicator media. And what it means is that if you have an organism that is growing using a particular metabolism, this means it could be growing anaerobically and fermenting. It could be growing aerobically, but maybe producing other compounds or other byproducts that are indicative of how it lives, what it uses, what nutrients it uses. This media will change color depending on the pH, how acidic or how alkaline the medium becomes while the organism is growing. So in particular, if you have an organism that is fermenting, and remember, in our tubes, we're going to inoculate. Well, we're going to inoculate all the way to the bottom of the tube. I'll show you in a minute. If we have an organism that grows by fermentation, it'll use the sugars, and this is called a triple sugar iron media. It'll use the sugars in here and convert them typically into organic acids, like lactate. So lactic acid bacteria, you produce a compound that becomes acidic. So if they grow by fermentation and produce acidic compounds, the media will turn yellow. If they don't, but they ferment in a different way, or that ferment, but they grow in a different way, the media changes in a different color. Actually, there's another compound in here called peptone. And when the organisms use that, the medium becomes actually more alkaline. So the pH goes up and the media will turn red. And there's actually a lot of different possible outcomes, because there's also iron and sulfate in here. And if the organisms can grow anaerobically, but instead of fermenting, they use sulfate as an electron acceptor, right? Because if there's no oxygen here, they'll use something else. And sulfate can be used as an electron acceptor instead of oxygen. they will produce hydrogen sulfide, H2S. That reacts with the iron that's in here, which will then turn black. So you can see that in this table, just to get an example on how these indicator medias work. So you can just review this and then get an idea on the different possibilities. So that being said, we'll just walk through how we inoculate these guys. And let me just get my board back here again. So you will need your tubes, your fermented food products. I only have one. I'm only going to demonstrate with this one, but you can repeat the same steps using each of the different food items. And you will need your inoculating pins. That's all you'll need. So the idea with the agar tubes, there we go, is that in our agar tubes, these are called agar slants. So they're poured such that you have a slanted region here. You have the media down here to the bottom of the tube. And then you have the surface of the media. So at the very top, this is, of course, oxygenated. But down here, because the media is covering this region, this is going to be anaerobic. So there shouldn't be any oxygen present. But, like I said, yeah, hard to draw upside down. There is yes. That is present. So organisms might be able to use this instead of oxygen, but then the media will be a different color. But there's also a lot of sugars in here too. So they can either ferment or do anaerobic respiration. This we'll find out when we inoculate. So when you inoculate, you'll have your food product. So let's say this is our yogurt. We'll just call it yog. You will take the pin that has the point on it and just put a little small dab. You'll drop it in and just a very small, tiny amount. Just cover the first, maybe like, I don't know, up to 3 millimeters on the very end of the pin. You will then take the pin and stab it directly into the media all the way to the bottom of the tube. So right in the middle point of the slant, in the middle of the agar, and then just drop it slowly in, push it slowly into the agar. Take it back out of the agar, nice and slow. I'm going to go through this. And then once you get to this part, back to the surface, just go back and forth a little bit, just to spread out the colony on top of the surface of the slant. So this will allow you to have one region, because you'll see the agar filling in as you're pulling the needle back out. This will have one region that stays anaerobic, and then the bacteria can then grow on the surface as well, and they will grow happily if it's aerobic, and they prefer aerobic conditions. Yeah? So let's just try that with our one fermented food product here. So we want to sterilize our bench again. Always a good habit. We're not going to have anything out, but it's always good to sterilize your bench in between. Sterilize your hands. Have our fermented food product, our pins, and our tubes. So let's take our first tube. You can hold it like this. And to pull the tube, the cap off, you just have to push gently. Just use your thumb a little bit. See how I'm kind of pushing it up? A little bit at a time until the cap gradually comes loose. Just have to pull it off a little bit here. There we go. There. Put the cap down like that. We have our tube. Take out one of your pins. There we go. And make sure that it stays sterile, so don't touch anything. Just hold it nice and careful in your hands. So now we have our tube and our pin. Open up your food product. And remember, this could be just this is just to represent any food product you have. It could be a container of yogurt, a bottle of yogi, a bottle of kombucha, but just this is our food product. You'll take the inoculating pin and just dab the very end of it, just a little bit. The first, you can probably see just to where the point comes to the, just to where the point ends. So right about there. Whoops, sorry, right about there. I'll even make a little mark. Just for demonstration's sake, don't do this, but just to give you an idea how much I'm inoculating it. There, I'm inoculating up to that blue mark. Because you don't need much, and you don't want too much of your food product on it, because if the food product's acidic, it might change the pH inadvertently of your agar. We want just enough to get the bacteria in there. So our tube, let me see if I can do this from this angle. So you should be able to see, yeah. I am now going to take and put the pin and stab it into the agar gently. There we go. And now you should see that the pin is all the way at the bottom of the tube. Just about there. Now I'm going to pull the pin really gently because we don't want to rip it out because it'll pull out a lot of agar and then we'll have an airspace. So we pull it out nice and slow, and you should see the agar closing around the pin as it comes back out. You might get a bubble or two in there. That's okay if it's near the top, but just so that near the bottom, it's closed. And then once we're at this point here on the slant, we're just going to go back and forth nice and easy. Don't scrape it too hard. Just rub the surface gently. Take it out. So this is now our dirty inoculating pin, and we don't want to touch that. The chemical's not harmful in this concentration, but we just want to be careful. Take this directly into your garbage and close it. And close it shut. You can feel it snap. It'll click when it's completely closed. So there's our food product. And I'll label that just FOOD. So you're going to want to label this with whatever you used. It could be the yogurt or the kombucha or the yeast. And there you have it. You can now repeat that with anywhere. You can use anything. You can use 4 different products. You can use up to 6. You have a couple extra tubes just in case. So inoculate with whatever number you want. And then that will be that exercise completed. If I were you, I would take these and store them in a small little container like this. So they're kind of right side up. And that is the culturing exercise. So once you have your skin cultures and your environmental samples and your agar slants, all this can be placed at room temperature in a darkened cabinet, even a little warmer, and let it grow for 24 hours. And then you can look at the results the next day. And in the exercise, you'll see that I have a few questions. I'll just put it down like this. So on the first, so this is day 1. So you have a few different hypotheses that you can check. So vilka organismer förväntar vi oss att växa på TSA Petri-skål eller på PDA-skål? So you can answer these questions here, just a couple. The next day for the observations, for the skin, the environmental samples, the soil and the water, you can look at this diagram to see the different variety of colonies that you might be growing on either of the plates. So for example, there might be some that are smooth in the entire colony, some that are called rhizoid, so they're a little bit more jagged, like a snowflake, some that have an irregular border around, some that are a bit more lumpy, lobate, and some that are very filamentous. The elevation is a bit more difficult to determine because remember, do not, oh yeah, that is one thing. After we've inoculated, do not open the plates. Keep them closed and we will observe them through the bottom part of the plate. That's just, because when you grow microbes in the environment, there might be a few cells floating around, of course, but here they can be in quite high concentrations. So we just want to keep them closed so nothing gets contaminated, or if there is always a slim chance, but it's still a chance to grow a pathogen or something that you might react with. So we don't want anyone to get sick. So just leave them closed. And when you're done observing, throw them into brännbart, and to burn them up. And then, and keep them out of reach of pets so no one eats it by accident. So you go through your chart and try to give an idea on the different colonies that you see. So here you can see, you can make a list of the different morphologies that you see on the different sample types for the different plates. And you'll see an example that I gave here. So maybe you found some smooth, round, a bit convex, larger than average, white, 2 millimeter. And in the soil, there were none on the PDA, but many on the TSA. In the water, there were many on the TSA, but none on the PDA. And on the skin, there was none on the PDA, but maybe a few on the TSA agar. And see how many different colonies you can find and give an idea on how many of each colony you find, of each colony morphology you find on the different samples. And then after that, you can answer a couple questions. You can then see how effective the disinfectant was. You might want to even give it a couple extra hours to see if anything grows, because the skin we noticed grew a little bit slower. So maybe instead of 24 hours, you could give it somewhere in the range of like 28 to 30 hours. And then you can see how different the colonies were, maybe between the skin and the environmental samples. So just make a few simple, basic observations. And then finally, with the tubes, you can see, write down what food items you used. If you use 6, just write it down under here. And then see what colors you get and what your interpretation might be based off of this chart. So to give some indication on to how the organisms from your different food items have grown. And then you can answer these questions here. And that is the extent of the exercise. You can go onto the Canvas website, and if you go to the gallery that's in the Digital Kursträff module, you'll see some pictures from what the other students have found. And you can use that and compare what you get to what they've seen in their observations. Great. that is the culturing exercise. And later in this video, immediately after this, I'm editing this video. I will include footage of going through the microscopy exercise, which you can use that to as a bit of a tutorial for working with your Foldscope. Okay, good luck and have fun.